Our next speaker will be Mahul Chakraborty, and he will be presenting Uncovering Hidden Genetic Variation Using Long Molecule Sequencing.
Good morning, everyone. So in last couple of days, we have seen talks or posters talking about mapping phenotypic variation using GWAS or QTL. They were using whole genome-wide SNPs or small indel genotypes to map the traits. And what they expected to find are SNPs or small indels explaining the phenotypic variation. But what they found, and which is a general trend in the literature, are SNPs which are not causal variants for the phenotypic changes. So where are the causal variants then? So that it prompted some people to come up with this crazy hypothesis that we are not finding the causal variants because they are harder to find. And what kind of variants are harder to find? So these are structural variants that include duplications, deletions, or insertions that are greater than 100 base pairs in length and cannot be detected with Illumina sequencing or microarrays accurately. So let me give you an example of what I mean by hidden variants. So this is a familiar story to most of you, the famous story of CYP6G1 gene, which is involved in natural variation in DD2 resistance. CYP6G1 has multiple alleles, all of which are structural variants. So it has one allele where it has an accord element and a duplication of the gene that increases DD2 resistance. Okay. Anyway, so we are going from left to right. Okay, so first with the duplication and the accord element, you increase the DD2 resistance by approximately 7-fold, and then you add this crazy incident that HMS Beagle, an 8 kb TE that inserts inside the accord element on the proximal copy, and you increase the DD2 resistance by a little more. Then you have a final allele where you have a P element insertion in the distal accord element, and that spikes the DD2 resistance to a really, really high value. So then the hypothesis is that there are structural variants, as you can see in CYP6G1, hidden in the genome, and we are not observing them. That's crazy, right? I didn't believe it, and I hope you don't believe it either. So let's see if we can find them. But finding those variants, the hidden variants, needs something extraordinary. We need to do a whole genome alignment to find all those hidden variants. But how do we find comprehensive variants using whole genome alignment? We have just one high-quality genome, that's a release 6. So where do you get a second release 6? It took 15 years to create a release 6 genome. We cannot wait for another 15 years to make another one. So what we did, we sequenced one of the 16 founder strains that are part of DSPR. For those who are not familiar with the DSPR lines, please look up King et al. 2012 or go to flyreels.org. It has more than 1,700 inbred lines that serves as an excellent mapping population. And the strain we sequenced, A4, is one of the founders that was used to generate these 1,700 lines. There is nothing extraordinary about A4. It is a cosmopolitan strain that was captured in Zimbabwe in 1960s. It's just a wild-type strain which has no artificially introduced structural variant in it. So fortunately for us, we didn't have to wait for another 15 years because we have PacBio long molecule sequencing. So this is how we generated the assembly for A4. We collected really high-quality genomic DNA which then was then used to generate 18.7 gigabits of long reads. 50% of that data is contained within 18 KB or longer reads. Then we created two different assemblies. One is only with PacBio only molecule using PBCR MHAP, and then another with PacBio only and paired end Illumina reads using DBG2OLC. And then we used a meta-assembly algorithm that I developed to merge these two assemblies to create a final assembly with 22.3 megabits N50. You can if you are doing PacBio assemblies, and you could use if you want to use my program, it's available freely on GitHub. So how does this amazing assembly look like in comparison to Release 6? So what I'm showing you here is a dot plot between Release 6 genome and A4 genome. The Release 6 genome is on the x-axis. You can see all the major chromosome arms, 3L, 3R, 2R, X, and 2L. I apologize for not orienting it based on their order. But you can see that in the boxed regions, which are the centromeric heterochromatin, is where the A4 genomes start to show fragmentation. But in most of the chromosome arms, which is euchromatic region, are in single contigs in A4. So in fact, the A4 genome is more contiguous than Release 6 genome, which has N50 of approximately 21.5 megabits. So if we have this amazing genome, now we can use this to align Release 6 genome and A4 genome and find copy number variations and other kind of structural variants. So I used another software pipeline that I developed called SVMU to call structural variants from this whole genome alignment. And what I'm showing you here is a distribution of just CNVs. I'm not showing you other stuff I have found, no TE insertion, nothing. These are only CNVs, the distribution of the CNVs across chromosome arms. And you can see that the parts which have been grayed, those are centromeric heterochromatin, and they're enriched with CNVs. The distribution of CNVs in the rest of the genome are more or less similar. But why? I mean, what is so important about this whole genome alignment? Couldn't we just detect this with some spatial algorithm and using paired and Illumina reads? I mean, they're also pretty useful, right? I mean, people have been doing that for quite some time. So to compare between what you can detect with state-of-the-art algorithms for detection of structural variants using paired end reads and what we can detect from this whole genome alignment, I'm going to show you the distribution of CNVs for chromosome 2L. I chose 2L just because it has the fewest CNVs, so it's easier for you to follow. So you can see that, so what I'm showing you here is the top one are the CNV calls from A4 assembly, and the second one is CNV calling strategy, or duplication calling strategy based on split read mapping, and the third one is based on read pair orientation. And one thing that's very prominent here is that we are missing more than 50% of the CNVs that are present in the genome, even with this state-of-the-art paired end-based technologies. But why do these things matter? I mean, we are missing some CNVs, but why do these CNVs matter? And where is the story about hidden variants? I haven't told you anything about hidden variants. So let's switch gear and talk about just one example. There are many examples I'm not going to talk about. I'm just going to give you one example. So this is based on an older story where Stuart McDonald's lab published a paper in 2014 for QTL resistance, QTL for nicotine resistance in flies. They used the DSPR lines to find 4 QTL peaks. Q1, I want you to look at Q1 and Q4 especially. The A4, fortunately, had high resistance allele for both of the peaks, Q1 and Q4. Q1 explains 6% of the variance in nicotine resistance, and Q4 explains 50% of the variance in nicotine resistance. So they found two candidate genes or candidate regions. One that underlie Q1 peak is Cyp28d1 and shows high expression difference between a low resistance allele and high resistance allele. And for Q4, they found an Ugt86D gene cluster that also shows high difference in expression between the high resistance and low resistance allele. So if you saw this result and then went back to your list of SNPs and small indels, you'll find all these SNPs. So what I'm showing you is the UCSC track for DSPR variants. And I mean, if I was doing it, I'll look at these SNPs and I'll see which ones are in the promoter region or UTRs, and I'll try to do CRISPR on them and come up with nothing. Because we are not seeing something with these variant calling strategies. And that is, so this, what I'm showing you again here are the CNV calls from 2L. And you can see that the A4SM, the genome alignment finds a duplication of Cyp28d1, which are not found by either of the strategies. So to look closely, this is what has really happened. So in A4, Cyp28d1 has been duplicated, and there is a 1.5 kb spacer between the 2 copies. Now, we don't know why there is such a huge difference in expression that caused by a single copy number increase. But it's possible that the duplication has removed a repressor binding site or some kind of other negative regulatory site. So that's for Q1. Now, what do we expect for Q4? I mean, of course, there is no duplication. I mean, it can't happen again. We have shown, for one, that there is this case. But, well, again, in Q4, there is another duplication of, but in this case, a Ugt86D gene, which is also involved in the detoxification pathway. And this is present in 3R. I didn't show you the distribution of 3R for, that you can detect with the different strategies for CNV detection using Illumina paired reads, but you can't detect this either. So you're missing 2 of the duplicates that are involved or that are really strong candidates for nicotine resistance, variation in nicotine resistance with paired end sequencing or any kind of variant calling. So I don't have much time to go over what other cool stuff I have found. So I'll just give you 2 examples that are representative of the insertion-deletion variants that I have seen. So if you go to Flybase and look at this gene, it has this T insertion between the 2 genes. And you can see this T insertion changes the UTR, 3' UTR length for these 2 genes. If you didn't have the A4 genome, you would think that this is present in all melanogaster, and it's represented by the reference strain. But this is the ISO1 specific event. And there are hundreds of events like this that are private to both ISO1 and A4. And lastly, this is another example where insertions are changing the length of introns. Here, what I'm showing you are 2 gene copies of the gene CYP309A2, and you can see this insertion of this T changes the length of this intron by a huge amount. So what's the future? I mean, do we sequence everything with expensive long molecule sequencing, or do we do better structure variant genotyping with short reads? So we have more platinum-grade Drosophila genomes coming for species divergence studies. And with that, I'd like to thank JJ Emerson. And you can follow JJ for more updates on platinum genomes, and Tony, and then the rest of the people in Long Lab and Emerson Lab. Thank you.